Welcome to this overview of malaria, where we trace the journey from parasite biology to practical prevention. In the next slides, we will cover transmission dynamics, clinical features, vulnerable populations, control tools like nets and drugs, and the research pipeline for vaccines and new therapies, all framed around actionable strategies to reduce disease and save lives. This slide summarizes the parasite species and the malaria life cycle in plain terms. 5 Plasmodium species infect humans, with P. falciparum causing the most severe disease, while others carry relapse potential. The mosquito injects sporozoites that reach the liver. Then, merozoites invade red blood cells, triggering cyclical fevers and onward transmission when gametocytes are picked up by another Anopheles. Here we look at vector ecology and what drives transmission. Anopheles mosquitoes typically feed dusk to dawn and breed in sunlit, stagnant freshwater. But behavior varies by region, with some species resting indoors and others outdoors. Control needs to account for insecticide susceptibility, habitat changes from urbanization and irrigation, and warming climates that expand mosquito ranges and risk to new populations. This page highlights who is hit hardest and why outbreaks happen. Young children, pregnant women, and people with compromised immunity face higher mortality and complications, while environmental and social factors like seasonal rainfall, zoonotic reservoirs in forested areas, migration, conflict zones, and overcrowded urban settlements all amplify transmission and vulnerability in local communities. Classic malaria presents with a recognizable clinical cycle: chills and rigors, followed by high fever and profuse sweating every 48 to 72 hours, which can mimic influenza, with headache, muscle aches, fatigue, and vomiting. Understanding this cyclical pattern helps clinicians suspect malaria early, diagnose it promptly, and start treatment before severe complications develop. Severe P. falciparum infection involves parasite cytoadherence and microvascular sequestration, which can lead to cerebral malaria, metabolic acidosis, profound anemia, and multi-organ failure. Even with proper therapy, mortality can reach the mid-teens %. So rapid triage, parenteral artesunate, and supportive intensive care are life-saving and critical to reduce long-term neurological sequelae. This slide lays out the core control strategies. Insecticide-treated nets have a proven impact, having child mortality when coverage is high, while indoor residual spraying complements nets but faces rising resistance. Looking ahead, larval source management, housing improvements, and innovative tools like attractive baits or gene-drive mosquitoes are being evaluated to sustain and broaden protection. Preventive interventions can dramatically reduce burden when targeted correctly. Seasonal malaria chemoprevention in Sahelian children, given monthly, can cut cases by around 75%, while intermittent preventive treatment in pregnancy prevents maternal anemia and low birth weight. Mass drug administration pilots in settings with resistance aim to interrupt transmission when paired with careful monitoring and context-specific planning. Artemisinin-based combination therapies are the current gold standard, delivering over 95% efficacy against uncomplicated falciparum where resistance is low. The 3-day combinations rapidly clear asexual parasites and reduce gametocytes, cutting transmission. But full adherence to the regimen is essential because partial treatment promotes resistance and undermines long-term effectiveness. Emerging artemisinin resistance, notably in parts of Southeast Asia, is linked to PFKelch13 mutations and manifests as delayed parasite clearance and rising partner drug failures. In response, research is advancing triple ACTs, synthetic endoperoxides, and PFATP4 inhibitors, while robust surveillance and containment strategies aim to halt spread and preserve the efficacy of existing drugs. Vaccine development is progressing on multiple fronts. WHO-recommended vaccines like RTSS and R21 have shown meaningful protection, reducing clinical malaria in children by roughly 30 to 75% in trials. While next-generation approaches, second-generation CSP constructs, multi-stage mRNA designs, and monoclonal antibodies seek higher efficacy, longer durability, and seasonal or pregnancy-specific formulations. Finally, achieving zero transmission requires integrated pathways combining chemo-vaccine strategies, sensitive rapid diagnostics, and real-time genomic surveillance to guide reactive, climate-informed interventions and cross-border collaboration. Above all, sustained political commitment and innovative financing are essential to scale these tools and work toward a malaria-free world within the coming decades.